Hello, everybody. Welcome once again to our OpenFlow cytometry live webinar. And today we're going to be carrying on looking at some data analysis, but obviously, first of all, a couple of housekeeping things. Hopefully you can see on the splash screen there that there are a couple of ways of getting in touch with us. You can use the chat function if there are any issues with the sound or anything like that. If you have any questions, and we certainly encourage questions, please put those into the Q&A box and we will try and address those as we go along. If there are any that we haven't addressed at the end, we'll address them at that point. Okay, so we are being recorded here today, so we should have a recording available as well, and we'll tell you where that's going to be at the end. As I say, we're going to look at flow cytometric data analysis today, following on from our first one about a month ago. I say we. Who are we? We should introduce ourselves. If you've been to these webinars before, you may be familiar with us, but my name is Derek Davis. I am the Science Technology Platform Training Lead at the Francis Cricket Institute here in nice and sunny London at the moment. And I'm joined by my colleagues from the US, so I'll hand over to Kathy. Introduce yourself.
Thank you, Derek. So hello, everyone. My name is Kathy Daniels, and I am the Flow Cytometry Core Facility Manager here at the Whitehead Institute in Cambridge, Massachusetts. I am aware that you cannot see me right now. I seem to have a video issue, but I am joining from a rather rainy Massachusetts today. So it seems that we've had a little bit of a swap, Derek. All right, and with that, I will pass it along to Rui.
Yes, hi everyone. My name is Rui Gardner. So I am the core director of the Flow Cytometry Core Facility at Memorial Sloan Kettering Cancer Center in sunny New York. I have to say that it is very sunny today and nice and warm. So things seem to be very glitchy today. Not only in my personal laptop, I had to switch to another computer because I had problems. And it seems that also the Zoom hasn't been working very well, and now Kathy is without video. But hopefully all these things will work out, and we'll be able to get along right.
Indeed, I think a combination of the sun and the rain and the internet are conspiring against us a little bit at the moment, but we'll see how we go. So what we're going to do today is sort of continue from last time. Last time, we looked at the basics of data analysis. And this is one of the things that those of us in core facilities have to deal with quite a lot. We train people how to run an experiment. We help them with experimental design and maybe even sourcing reagents and sample prep and so on. But quite often we let them go off with their data to play with it. And they should come back to us, but they don't always do that. And sometimes people don't develop the level of confidence that we'd like them to. So one of the things that we wanted to address with some of these webinars was to give you the confidence in the basics of data analysis. So last time we looked at FlowJo, and we looked at the sort of anatomy of a flow cytometry standard file. If you remember, all flow cytometers will produce data in the same format, flow cytometry standard format. We looked at the sort of simple ways we can display our data by using polygons and different types of bivariate plots, and the ways that we always have to reduce our data to the things that we're interested in. And we do that by using regions and gates. And we also looked a little bit about the data scaling that we can change in the analysis portion. So we'll probably start today with something very similar. Just get you used to a slightly different software and maybe a slightly different way of doing things. But last time we did a couple of very simple experiments. We just looked at a basic viability experiment and a titration experiment. Today we'll up it a little bit, and we're going to do a phenotyping experiment. Not hugely complex. There's there are 6 colors in there. So we'll be looking at the way we analyze that data, the sort of gating tree, and hopefully we'll also be able to cover the use of controls, particularly the FMO, the fluorescence -1 control. And if we don't run out of time, we might look at some of the outputs, the sort of batch processing that we can do And as we've said, questions are definitely encouraged along the way. So today we're going to be using FCS Express, and Rui is going to be running this. And as he said, his computer may be a little slow, so we might have to swap between his and Kathy's. We'll see how we get on. Rui, I hope you're still there. I'm going to hand over to you. I'm going to stop my share.
Yes, I am here. And so this is going to be fun today, but I hope everyone can bear a little bit. I mean, in a way, it's good to also do things slowly, because then you can actually see it better. Usually when you have those people doing those, they're really proficient and doing all those demos so fast, then you end up missing a lot of details. So hopefully we can do this rather slowly. Yeah, I'm running into, again, several issues, but we'll try to improvise here a little bit. So I'm going to share my desktop and start with so usually FCS Express starts with a pop-up window, which is quite handy, and this time it's just refusing to start with that window. And I know we can see it somehow. Oh, here we go, startup screen. So usually it starts like this, and this is quite convenient, because you can actually look at you have links to some of their videos. If I click here, we'll go to some of the tutorials that they have, so you can browse this and find out more about the software. Actually, I'm logged in, so if you have an account, actually now to use FCS Express, you need to have an online account. And so if you go into your account, it might not be very obvious because you don't see it here in the top menu. But if you click on your own icon, you'll see that we have downloads and videos. So for instance, if you click on videos, you'll have a lot of tutorials of FCS Express, helping with from basic to advanced analysis. So quite comprehensive. A little bit of publicity here. So if you go to our website, actually, and you go to education and training, you will see that in the analysis software, we have a series of FCS Express classrooms that we did last year, exactly, during the lockdown and during the pandemic. And we also have here links for the videos, tutorials, and manuals. So a lot of resources here to, if you want to find out more about the software. Here it's a bit, the objective here is to do kind of both things, show you a different software than we did last time, but also to, what we really want to show is how to do data analysis, and it should be independent of your acquisition or analysis software, right? So again, like I said, once we typically start with this window, and then we just click new layout, which I'm already there, and it will open up something like this, right? A blank page. Now there are several ways. Oh, you want to say something, Derek?
Yeah, sorry, I was going to ask you a question, Rory. So you're running this on a Mac, obviously there. So yeah, this, like FlowJo, will run on a Mac or a PC.
Exactly.
Similarly.
Exactly. So it runs exactly the same on a PC and the Mac. I have to say I had more experience with it on a PC, and I think the Mac version probably is not as stable as one would wish, but it still works pretty well. The big difference between the PC and the Mac that I would say is, with the PC you can actually just copy paste the plots, the objects to PowerPoint, and then you can ungroup and convert it to a PowerPoint object, which is quite nice. So then you can modify the titles, the fonts, everything in PowerPoint. But with the Mac, you actually have to do it within FCS Express, and then you can export as an image, right? So I think that's the main difference. And I'll show you, hopefully, with the batch processing, I'll show you how to export everything to a PDF or to a PowerPoint slide. which is one of the features that FCS Express has very nicely, is that you can actually export it to these kind of softwares, right? To to share your data.
So we do have a question already, actually, which I imagine we'll address as we go along. But what is the benefit of using FCS Express compared to FlowJo?
Yeah,
and I suppose that depends on where you are and who you are.
Exactly. So, a lot of, I think mostly because at least at the basic, all the things we're going to do here at the basic level. And when I say the basic level, probably 98% of people doing flow cytometry analysis are only going to cover the essential, the basic functionalities of flow cytometry analysis. At that level, I would say both software just work very well. It might be just a question of preference. The nice thing with FCS Express is that it has this ribbon, which is very similar to PowerPoint. They even have the color of PowerPoint. So it's almost like you have your worksheet here where all your plots are going to be, placed, and you'll see that it's very familiar if you're familiar with Microsoft. And in specific Microsoft PowerPoint, but again, in terms of functionalities, both work very well. Usually, I recommend if you have people in your lab that are already using FlowJo, maybe you're going to get more help and more support, more immediate support. If they have already FCS Express, maybe using FCS Express is a good idea because you'll have more immediate support. But having said that, both companies are very responsive. If you have any issues, you can always reach out to them or your core facility. I would say it's pretty much the same.
Yeah, exactly. I would say most big core facilities will probably have copies of both softwares, won't they? And you would direct people to whichever one they're most familiar with, or whichever one does something slightly different. We'll see. We'll see some of these things as we go along, I think.
Yeah, when we go to more advanced analysis, then there are going to be some differences. So maybe one software will be better than the other, and vice versa, depending on what you're looking at, because those are the tools that they keep implementing, and as these tools also keep coming out and being published, it depends. One software will be faster to implement than the other, so it really depends. But I would say mostly it's the same thing. Okay.
Back to you.
Okay. So there are, just like FlowJo, FCS Express, there are several ways to load the data. So I have here, a folder which has loads of files. You have the compensation controls, the FMO controls, the samples. Here today, we're not going to really do compensation unless we have time and you want to go through it. But we'll already load the data compensated, and so we'll look at more of the aspects of how to analyze and gate the data and look at statistics and things like that. So as I said, there are several ways. You could go to file, load data, etc. I like just to, I pick my sample and I just drag it, I drop it, and there we go. It asks me, okay, do I want to open as a dot plot, density plot, color dot plot? I usually like to just load the density plot. You can click here to remember the selection. It's up to you. So if I do okay, then there we go. It loads the plot. Now, where's my data, right? Where is my data file? If you want to know more about that. The way to do it is you go here and to data. So this is one of the nice things in FCS Express. It's very intuitive, right? If you want to insert a plot, you go to insert. You have all these different types of plots. If you want to do something about the design, the layout of your worksheet, you go to design. So gating, all the functions to gate will be in the gating menu. So it's relatively intuitive. And so if you want to look at your data, then you just go to the data menu, and then you click data list, and it will pop up. Okay, let me click again. There we go. It's slow, but not that slow. Sometimes, and this is something I noticed with the Mac versus the PC version, is that sometimes I click on things and it seems not responsive. Then you click again and it responds. So that's something I've noticed. But okay, so the nice thing is that I loaded only one file, but it automatically loads all of the files in that folder, right? So this is quite nice. And then you can, instead of having it floating around, you can use these buttons to place it on one side, for instance. I rather do that because I have here a lot of real estate. Hopefully I can increase the size, zoom it a little bit if you need, if everything is too small. But notice that here we have all these files. And as I said, we're not going to really look at compensation. So I rather just remove these files. And so I just select them all, remove. Now, don't be afraid, because although I've deleted them, I haven't deleted them from the main folder, right? Everything is still here. It just deleted them from our workplace. So it's not really deleting it from the hard drive. So now we have the FMOs and the samples, so we can start, maybe exploring the data. I know that one thing that people like to look, and that's actually important, is to look a bit at the header. If they want to see information of the file. So if we click here, data, view header, we can see a lot of information about our file, right? And the file name, the operating system, the names of the parameters which are important. The values are all here in this table, and it has also things like all the information that you would see in the header, the voltages that were used, so the PMT voltages in this case of this instrument, and it was this is data from our instruments here at MSK, and so it has a lot of the information that you will see in the header.
Okay, I can ask a question there, Roy. Does it have the compensation information? Maybe there was no compensation set in this file, but
maybe there was no compensation matrix. That's a good question. Let me see. Where do I see that? Because you are supposed to see that in the data. No, yeah, now I forget what it is, but maybe Kathy can look quickly and see if she finds where it is. Oh, here we go.
Yes.
Transformation.
So if you click on compensation and unmixing,
Right.
it'll show you what was automatically loaded with the data file. So that specific file might not have one, but if you click on one of the other ones and you expand out on the automatically with data files,
There it is.
right there, it'll absolutely show you the compensation matrix and all the associated values.
That came with a file, right?
Yep.
Exactly. So this is the compensation that the compensation matrix that was recorded with the file while doing acquisition. And so this is something that's always possible to see. So like Kathy was saying, for instance, this sample that I happen to be starting with doesn't have the compensation matrix, right? But all the others seem to have, as we can see here, right? This would be, and if I click here, we have the compensation matrix with all the values that are being applied, right?
Yeah.
Yeah, so very likely.
Everything, it's a question of remembering where those things are.
Exactly.
It's easy enough to get them.
Exactly. And again, I knew that there was a way to, right, to show that, because these are the things that we need to expect. If a software doesn't have access to that, then there's something wrong with the software. That we should be able to see these things. We shouldn't be able to tweak them. That's a different thing. Like I just did this here. But anyway, we'll actually I'll actually show you a good case where where you shouldn't, not where you shouldn't ever, but I'll show you a case where it's obvious, where you could really mess up your data by tweaking compensation by hand. Okay, so let's start doing the analysis now. One of the things we always try to convey is what is the objective of this analysis. You want to identify populations, and the first thing really, or most of the analysis is really to clean up the data, right? To remove debris, remove dead cells, and just make sure you're identifying specifically your populations and not anything else, right? So you can start in any order. You can start with the singlets, so the doublets exclusion. You can start by the debris removal. You can start by the live dead. It's really up to you. There's no right way to do it. Oh, sorry, no, they're all right ways to do it, but there isn't a single right way to do it. That's what I mean. Okay, so I'm going to start here with, let's say with, just a forward side scatter. Oops. Let me see. Actually, I'll start with that's why I wanted to go to this file here. So this is actually a nice example where, oh, by the way, I didn't explain these samples here. If we click here, we can change from sample to sample, right? It will refresh the data. I'm going to start with the live dead, but notice that during acquisition, unfortunately, there weren't any labels, and so we basically, the person that acquired this didn't write the labels, and so we only have the fluorochrome list, right, or the name of the channel. So it might not even be the right fluorochrome that's being used, but it's just the name of the channel, right? And actually, in this file, we do have the names of, so it was labeled, and this is nice because now we can see better that, okay, we have Ki67, CD44, CD25. It would be nice to see both, right? Which is possible, but at least like this, it helps us during the analysis because you may not remember, and even for this demo, it's easier for you to remember what's a CD4 and CD8 than what APC or APC size 7 is labeling, right? So we'll start with this file and then we'll explore everything else. So we can start with side scatter here, and then let's go for instance for the live dead. And as you see, what we have here is we expect, and this is again, the data analysis in flow cytometry is very iterative, right? We're going to start by selecting what we believe is our population based on our prior knowledge. If you don't have any knowledge, there are ways, and I'll show you a little bit how to do that. There are ways to figure out what are these, all of these populations that are showing up here, right? So with the live dead, we expect to have cells that are negative for the live dead, which would be the live cells, and then everything positive should be dead. But we may have different populations with different autofluorescences, right? So for instance, this population may not be positive. It may be just more autofluorescence than these populations, right? So we'll have to go back and figure out if we're gating out the right populations. So okay, let's start with the gating. I'll start with a polygon, and we can select the negative. Since I'm looking what would be the most likely the granulocytes or monocytes over here, the and they're usually much more autofluorescence. I am assuming that these are already cells that are labeled, right? Because if they were highly autofluorescent and not labeled, they would probably be have here a much more higher side scatter because these are PBMCs. So I'll call these cells, and I'll leave the color in red. Right, so we've identified this first population. Let me just bring this here so you can see, and immediately, by default, FCS Express will show you the percentage, which makes a lot of sense. And now you can, you're probably asking, why are you cutting this like this? Maybe you're getting a little bit of these, etc. Well, I'm not too concerned about that right now, because I want to see how the other plots look like. But I definitely want to. I definitely want to make sure that when I create the other plots, I'm not losing out cells. So we'll play around with the gates. So for instance, I will create, so I just drag and it opens up the data within that gate. So now actually, yeah, we can do something like, well, side scatter hides. Actually, I already had side scatter area. and do it like this, right? So now I have my doublet exclusion. As you see, it follows a diagonal because we're plotting height versus area. These are most likely aggregates. Now, are they really aggregates? We actually have here an activation marker. Be aware that sometimes when cells are activated, for instance, B cells or T cells, they actually increase in size. And so they will show up somewhat in the doublet side. However, they usually also follow the diagonal, right? So in principle, these will be doublets, but we can try and see later on if this matches, based on all the other samples also, to be able to really exclude them or not. So I'm going to do righty. Okay. So I'm going to start drawing, and now one of the things that is important to to understand when you're doing these kind of gates is you want to be as objective as possible. When it comes to doublet discrimination, this is probably one of the most difficult plots to gate because there isn't much criteria, right? There isn't a control where you have all singlets and then a control where you don't have singlets. We wish if we could have everything singlets. That's the objective, is to have as much as possible a good singlet, single cell suspension. So we don't have that control, so we have to base ourselves on the knowledge of the voltage pulses, etc. We won't go into details here, but essentially you want to use this gate and follow the diagonal in which the populations are going in, right? The distribution. And that's basically the criteria. Don't try to do a polygon and go around them. Just try to follow, follow as much as possible the diagonal that's showing here.
Rui, is it okay if I ask a question actually?
Yes, sorry.
Okay, no, it's okay. So I have a quick question for you. So, you're talking about the activation and the ability to pull out single cells and how it might change based on activation status. And for me, that brings up the point of different samples might need slightly different gating. So does FCS Express allow you to have sample specific gating, or does that same gate apply to all the samples?
Right. Yes, it does. In fact, if you go to gating, you see here the data specific gate, right? As we select the gate, I could click here, data specific gate. So let me just show you, if I refresh the data, the gates are all the same, right? They're being applied equally. And if I click now here on this gate, and I select data specific gate, and let's say I move it, let's put it over here, just to be obvious, right, that it's in a different place. Now, if I click on another file, I'll see that it still has the original gate. So all of these have the original gate, except this one that will have the gate moved. So we can do an analysis having gates that are specific for that file.
Great. And then we did have one other question from Nagwa that came in asking about if the sample that you're gating on is the unstained sample right now.
If the gate, no, I'm gating on one of the samples, right? So I'm going to do first an analysis, based on the scatter, singlets, etc. And then when we start gating the actual populations, we're going to use our controls to be able to set the gates appropriately. Okay, and if we have questions like, for instance, what are these, are these cells labeled with something? We can look at the unstained and things like that and see if these, compare the unstained, the FMOs and the data, and see if this population is really, for instance, an activated, a population, right, of cells.
Sounds good.
Mm-hmm. So I'll just eliminate here and let me see. Right. Okay. So now I drag this, and what can I do here? We've excluded the dead cells. You know, one of the nice things of the scatter plots is that we can see whether you know, cells seem to be healthy or not. For instance, if you see a lot of debris and things squashed in the corner, I'm just going to look at the scatter plot here. Sorry. And, you know, as you see, we don't see much here in terms of things being squashed in the axis. And, you know, it does seem that cells are, you know, relatively healthy. You know, the forward scatter could have been increased. These are the kind of things that, you know, one starts learning. And, you know, the reason why we're also using these kind of data is to show you things that actually happen in reality, not only necessarily, you know, data that's perfect. And you can see that, for instance, here we start seeing things that are a little bit more squashed in the axis. So we can't really see whether the data, whether the cells, whether we have a lot of debris or not, right? So here we don't have really a way to exclude debris. We'll have to go to the, you know, and gate on our populations, and all the populations that are labeled will not be debris, right? Okay. So here I'll just you know, skip immediately to let's say CD8 versus CD4, right? So we'll look at only the you know, the lymphocytes, and here, although we have, as you see, a lot of other markers, for simplicity, we'll just look at the T-regs. Now, for instance, remember I was saying, you know, these could be monocytes or granulocytes. You know, we don't know, or maybe these are all the monocytes that are here or something like that, right? So if you're in doubt, one of the things we can do is use backgating, right? We can use the markers that we added to be able to go back and check to see if we're, you know, really excluding the right cells, or if we're actually losing some of the populations of interest. So one of the things we can do, for instance, we have here a granulocyte marker, right? So I'll just I'll put this here like this, and I can see that, yeah, CD8. Let me see if we can use another marker here of yeah, like CD45, right? Well, actually, we don't see it as much, but we do see that we have. I'm going to put CD8 again. Because these will be the lymphocytes, and these will most likely be the you know, granulocytes. So we could start exploring this data and do some kind of backgating, and I'll actually I'll show you probably more backgating as we go along, to try to just identify, to understand what these populations are. So the way to do backgating is we start by gating cells that we're sure, you know, what they are. Right? Actually, you know, okay, oops. Let me go back to them, to the CD4. And, you know, I'll gate here on the CD4 positive or even the CD8 positive, and just to show you where the lymphocytes are, right? So if I draw the gate here, these will be for sure CD8. So I'll just call them, you know, oops, CD8. Actually, for now, I'll just call them backgate. Okay, because I'm going to use this as a backgate. And the way we do it is, we don't want it to be a child gate of anything. We want to see within all of the cells. So I'm not going to put a parent gate. So this is going to be looking at all of the cells that have that signal, right? So we should be mostly enriching it with CD8s, right? And now I'm going to convert this into so I go here to format. I convert it into a color dot plot. And the nice thing here is that FCS Express. I should change the color of the dot plot, because this one is also blue, right? So we don't really see where we don't know if it's this gate or this gate. The way to do it is we go to insert, and we have here the gate view, and I can draw, and I have here my gating hierarchy, right? So these are the cells, singlets, and you see they have both the same color. So actually I'm going to double click this and change the color of this one to green, right? Oops. Let's see, more green. I know for some of you this may seem green. For some it may seem blue. So there you go. Now we see much better where the green cells fall, and I can update this in real time. So if I go here, we see that, oh, these are mostly these cells that, as I said, could be granulocytes or monocytes if you have, if you're in doubt. But I can see that the lymphocytes are mostly here. And if I gate the CD4, I see that a lot of the lymphocytes are here. But it seems that CD4 are starting to, label as dead cells, right? So again, looking at this, I think we're gating appropriately. I still have some questions here whether these events here are live cells or are just or could be dead cells. But I suspect since CD8 and CD4 should be roughly in the same region, that CD4 T cells are starting to die. These are definitely dead cells, right? So it could be up to you to gate these or not, but as long as you do this systematically to all your samples, even if there's an error, this will be a systematic error, right? So this helps us to start interpret, okay, these guys here are mostly those over there. So so now if oops, it's a bit annoying. If we go to the GR1 or granzyme, actually this is granzyme B, right? And actually I thought I was interpreting this as GR1, so we actually don't have a granulocyte marker. But anyway, yes, this is only for lymphocytes. Okay, but you see the power of backgating, right? Now we can even leave this like this, and let's go back to the CD4. Actually, there we go. And I can click here and just, oops, click the wrong, delete. There we go, delete. And we get rid of this, right? So now we have CD8 and CD4, right? So if I go to gating, here the question would be, okay, these seem to be positive and all these seem to be negative, right? I really don't know because we need an FMO control for this. So, you can start by setting a gate like this. Let's call these CD8 positive, and this is important. So CD8 positive, let's change the color, I don't know, to pink. And then, we usually like to just extend this gate as much as possible, because you're trying to find all of the cells that are positive for CD8 and negative for CD4, right? So they're not the double positive. Yes, Derek.
No, you're probably going to go on to this actually, Rory, but I was just wondering whether everybody knows what an FMO is.
Okay, right, good. Yes, so the FMO stands for fluorescence -1, which basically means that you label with everything except the fluorochrome that you want to set the threshold between negative and positive, right? So in this case, I don't know exactly where to set the gate for CD8 and even for CD4. I mean, it may seem obvious that for the CD4 is here, for the CD8 is here, right? So without any controls, I would set the gate here, right? Now I want to look at the FMO for CD8, which would be adding all of the antibodies, all of the fluorochromes, except the CD8, right? And so, and now I'm going to look for the FMO, because again, the person that did this didn't label, so we don't know which FMO we really need. But okay, I'll just start. I know that 10 is not, so I'll pick the 9. It's not. The 8 is also not. the 7. So I'm looking for when the CD8 signal, which is for CPSI 5.5, by the way, disappears, right? So I go, of course, if you had it labeled, you wouldn't have. Mhm.
I think it might be FMO9, if I'm correct on this
Actually,
one.
I think
Sorry, FMO4, sorry.
Looks like that one, doesn't it?
yeah. But I'm
Yeah,
also
I
yeah.
was going to say, I suppose one thing that we've said we'll look at next time is compensation. And when we compensate, we have single color controls. And the idea of a single color control is that we see where the photons from that particular fluorochrome end up, and then we can account for them by the process of compensation. And the FMO control is almost the inverse of that. So we would have every color except the one that we're interested in, and that then accounts for photons from other fluorochromes in the system that are ending up in that particular channel. So the FMO is an extremely important control in multicolor staining, especially where, as we've seen, if you've got obvious positive and negatives, you may not need them. But if you've got that continuous expression that we quite often see, especially with those sort of markers that are modulated during expression, then an FMO is very important.
Exactly. Yes. Thanks, Derek. I forgot to mention that, and that's probably the most important point in using the FMO, is that we're accounting for all of the spillover from all of the other fluorochromes that will increase the spread. So notice that there is a lot of spread here. Actually, if you're not comfortable, and this is something that we can also look at at scaling. If you're not very comfortable because there's a lot of space here, and you want to see this in a more logarithmic like, there are ways to do this. You can just in this case we right click format, and I need to take the video from the way, and so here we want to adjust the scale in the x-axis. So we just click on the x-axis, and here you have the bi-exponential that's being displayed automatically. So you can just remove that and adjust this nicely in a manual way, right? And so now you see that, okay, they're more, some people prefer to see it like this. It's more recognizable. They feel more comfortable with that. Looks nicer. But really, what we're doing is just changing the scaling. So the data is still the same. Notice that the gates adjust also. So nothing changes. It's just the way we visualize the data. So you're not, in this case, manipulating or changing the data, right? You do have to be careful, because a lot of these things that we do, we tend to recognize patterns, etc. And by changing the scale, you may not see some of the patterns that you're used to. So be careful with that. But you can always play around with the scaling and get more accustomed. But it's not crucial. So the way it was, I would be okay with it. It doesn't really matter. It doesn't, for me at least, it doesn't really help that much. But some people find it much more comfortable. Oops. So I can go again and do it like this, right? But from data to data, we might, a different scaling might be more interesting. So here we have the FMO for the CD8. Remember, the CD8 is on the y-axis. Notice that I set the gate here because I thought that the difference between the negative and the positive, because there was a lot of spread. So let's go back to, I believe I was here, right? I was setting the gate here. I thought that this was just the spread from the other fluorochromes, but it happens that it's not. It's not the spread from the other fluorochromes, because when we went to the FMO control for CD8, actually this is the spread that we're getting from the other fluorochromes. So one other thing that we forgot to mention also about the FMOs is the FMOs have to be the same sample, right? We've seen people sometimes bring, just to save their precious sample, setting FMO controls in another tissue. But as Derek was explaining, we want to take into account all the photons that are coming from the other fluorochromes. And so these depend on the levels of expression of the other markers, right, that are labeled with a fluorescent probe. So to do so, we need to label exactly the same sample that we are trying to compare with. So that's very important. And sometimes it's very difficult. We've discussed this in other sessions. You know, what if you have a clinical 100 clinical samples from 100 from 100 patients? Do you have to do an FMO on all of those patients? Well, ideally, yes. Unfortunately, and for practical reasons, it's not possible to do that. So you know, you end up you know, sacrificing a little bit of the you know, the quality of the data. Hopefully, you know, in most of the cases it won't change that much. And for instance, I see here that CD4, it's pretty obvious where the negative versus positive is, so we don't really, maybe don't need an FMO control. But you know, for some markers it may be, you know, it's what it is. So it's not perfect, but it's at least the best control for that. So so we adjust. So I adjust it here, right? To, you know, to set it close to the negative, because these are negative. We didn't label it with CD8, but I still don't know how much to go to the right side. I need the FMO control of the, so when are these double positive? When do the double positive start, right? So we need the FMO control for the CD4, which I think I saw it somewhere. Also, oh, there it is. Oops, there it is. So the FMO for CD4. So I'm seeing here that, oh, okay, it's more or less here, correct? Right. And now I'll set the gate for CD4, and I'll just do something like this.
So I'm back again, Rui.
Yeah, go ahead.
I think one of the things that, particularly when you first start off, you know, we like to think of flow cytometry as this fantastic, almost completely objective technique. But even though we have an FMO, which is the right control for setting that cutoff, you know, the question that I would get asked is, you know, why have you set it there? Why haven't you set it 2 or 3 pixels higher or 2 or 3 pixels lower?
Mm-hmm.
So there's still a little bit of subjectivity in there, isn't there?
Exactly. Yeah, so correct, and you know, you saw that I was there, you know, hesitating a little bit. Now the, you know, one of the criteria that we typically use is, you know, also depending on the you know, if you have a relatively frequent population, you know that 0.34 is anything but below 1%. Will be negligible, right? So so the thing is, I could get it closer, but as long as with my FMO I have anything below 1%, of course, if the percentage of population that you're expecting here is going to be at the level of 1%, then 1% for the FMO is too high, right? To set the gate. You want to have something maybe in below 10% of that percentage, right? So in this case, 0.1% would be something, you know, or less.
Exactly. I suppose you have to apply a bit of reality to this as well, don't you? I mean, you could set that marker so that there are absolutely no dots above it, couldn't you? But you might end up setting that in a completely unrealistic position in that case. Or you could, as you say, set it to always be 0.1% or 0.5% or something like that. But even that's not ideal. As you say, if you're looking for a rare event, that's going to confound your statistics a bit, isn't it?
Exactly.
And your belief in that result.
And I'm going to jump in and just add another little comment here. Sorry, my video is not working. But this also assumes that all of these reagents have been appropriately titrated as well, and everything has been optimized for this specific panel, right? So something might occur where maybe you're adding a little bit too much antibody at the manufacturer's suggestion without titrating, and then the negative can shift up. So all of these controls are dependent on the experimental optimization that goes into the panel setup beforehand.
Right. Yeah, that's absolutely, you know, that's an extremely important point, because, you know, a lot of the, you know, problems and the frustrations that people face when they're doing these and the questions that come up when they're doing this kind of analysis actually end up being not a problem with the analysis that needs to be done in a different way, but more because the sample preparation, right? Everything coming up to the acquisition, and even sometimes during acquisition, that wasn't consistent or wasn't done correctly, right? And then, of course, then you run into problems like Kathy was saying. If you don't titrate well your antibodies, you're not going to have a very good resolution. And for instance, you may end up with something like this. And you don't know then if this is something to expect biologically, or if it's just an artifact of, bad sample prep, right?
Exactly. I think there's a question there. I don't know if you were addressing that, Kathy, or I was slightly here. Looking the other way about the fact that you get a slightly more spread there in the negatives, but that could come from multiple sources, couldn't it? It could be that there is actually really some low-level staining in there. Mm-hmm. It could be that maybe you're getting a bit of background staining, but as Kathy was saying, this should have been optimized in terms of titration. Yeah. Or it could be that you've rushed your last wash as well, isn't it? That right. You've got some free antibody knocking about, which is contributing to background as well. So there are several. You have to be sure that it's a biological effect and not something that you've introduced into it.
Exactly.
Yeah, absolutely. And that's even other things like making sure that you use an FC block before you carry out your staining, or making sure if you're using a secondary alone, which, I don't suggest in general anyway, but making sure you spin that secondary alone down and just use a supernatant so that you're not getting any really sticky secondary that's non-specifically binding. So all things like that to optimize will help you really be certain if it is low level staining that you're seeing that you can gate on and be certain about, or is it just some background that you're seeing?
Right. So okay, so I went ahead and placed the CD25 versus FOXP3. So we want to identify our T-regs, which would be double positive for CD25 and FOXP3, right? And now, the interesting thing is we're obtaining here a population. Actually, even before that, this is a spleen. And if you know a little bit of biology, we are capturing some CD8, CD4 double positive cells, right? Which would be a bit strange for a spleen, right? If this was thymus, it would make sense. Actually, it would make sense to have many more there. But this should already be, if you know a little bit of the biology, it starts giving you also, a red flag. So what is this? Why am I seeing double positives? Maybe I haven't gated out all of the doublets here appropriately, or maybe it's something else. So again, using backgating, let me just take this out of here. Using backgating is actually quite appropriate. So again, let's try and see where this population is. So I'll do the same thing again. I come here, I draw a gate just over this, and again, let's give it a nice different color. I think we were using green or sea green. Oh, and let's remove the parent. And so you just click OK, and do you see? It's essentially this population, right? You have a few of them there. Maybe it's these these events here, but the main population really is this one here. And so if you're interested in just the lymphocytes, then you can leave it there because we're gating them out. Here we're getting the CD4 positive and then looking at the CD4 positive, so we're excluding them anyway. But if you feel more comfortable, and for instance, when you're trying to show and present this data, whether it's in a lab meeting to your peers or even as a manuscript and publishing it, you may want to clean up that. And so, for instance, we could come here and we could use either this gate or even this gate and just reduce it. No, what am I doing? Oops. It's just complaining that my computer is slow, and so we could just let's just do this, and and remove, and you see that now we don't have that population there. So you could do it like that. Again, I wouldn't be too concerned about that because again, we're excluding, this is not a population of interest in this case. So we're eliminating it in the gating strategy anyway. But again, if you feel more comfortable, because then, maybe some people would question, oh, this is a spleen, what are these double positives doing here? And you would have to explain, no, these are the granulocytes, or the monocytes, forget it. So it's a nice way of just, eliminating those, gating out those populations as you're going along. Again, as long as you know what you're doing and you're gating them appropriately with the right controls. Okay, so again, as I was saying, so we have here a population. Is this negatives plus positives are all together? What's going on? We'll have to use the FMO controls. Looking at the other samples, we'll see some differences. So here, do you see that, for instance, this gate of the CD8, CD4, that we're still capturing a little bit of the tail. Now, is this relevant or not? You can check, right, to see, let's say if I would pull the gate, so it's 14.62, and if I go over here and eliminate that, that's 14.24. Is it a big deal? Again, within your the percentage of the frequency that you're expecting, no, it's not going to be a big deal, but it could be, right? So you may want to look at these things and try to go back and adjust and make sure that these gates are appropriately set, right? I'll just, actually, the nice thing here is that we can undo and just place it back, right?
Yeah, I think you're right, Rui. You don't want to get too hung up on a tiny difference like that. If you've got a big percentage, it's really not going to make a lot of difference. And also, you're never going to be doing this experiment once, are you? So you're going to have a range. In fact, if you rerun this sample, you'll get a slightly different percentage in there. So we always have to realize that there's always going to be variability. Some of it is biological, some of it is what we do to the samples as well, and it's what we're prepared to put up with. But while we're at this point, there's another couple of questions here, and before we get too far away from the FMO, there's a question here asking, could you use, if you've got those 100 clinical samples, could you take a bit out of each of them and use that as a sort of combined FMO?
Yeah, I mean, it's one of those things. Then you'll just get we've discussed this in other circumstances, like the autofluorescence when you're doing unmixing, and without going into drifting away too much, but it's one of those things that, well, if you gate just everything and you take into account the average levels, I mean, not really, because all your samples will be skewed, right, towards whatever. I mean, if most of the samples actually are the same and have the same levels of expression, maybe that will be okay. But if there's a huge variability, then no, it will be. Yeah,
exactly. I think if you've got the odd outlier, you're in a bit of trouble, and it's maybe a compromise too far to do that.
Yeah. In that case, I would say it's really, it doesn't really matter if you're using, you try to use a tissue that's as similar as possible, but still, in that case, yeah, it will be difficult to have the right FMOs, and that's why there it's also very good to have things very well titrated and make sure that you're pulling out the populations as well as possible. Because we have one of the things we have to understand is that these are the limitations of this technique, right? And we have to be careful not to abuse, especially because there is some subjectivity there. And so what I usually say is that whatever you do, wherever you set the gates, you always need to use some objective criteria, right? So always, when you place a gate, always ask why, for instance, in this case here, why am I drawing the gate like this and not drawing the gates, for instance, including more of these guys, right? Now, notice that I did this. Did I change much in the percentage? Nothing changed. If you start, and that's one of the nice things of this software, is that you see things change in real time. If you move around your gates, you see that the percentages basically remain the same. So you can be comfortable that, oh, okay, I don't have to be so picky, with these gates, because it's not really changing my results. Having said this, this is
that
just
there
one sample, right?
Yeah, there are 60 or something people on this call. I think if we gave them all that data file, it wouldn't, none of those gates should be in exactly the same place, but we would all come up with approximately the same answer.
Exactly. Exactly.
Okay, so there's another question here. Sorry, Kathy, about do we need an FMO every day or every time we do it? My
Yeah.
answer to that would be yes. Yeah,
Exactly.
quite a simple one there.
Exactly. There are many reasons for that. I mean, the instruments drift, the reagents drift, especially if you're using tandem dyes. You can't expect, from experiment to experiment that those tandem dyes will be exactly the same and have the same spectral properties. And the same thing with the single stain controls for compensation or mixing. You should bring them always fresh, or even better. You should always stain them at the same time as you're staining your sample, if you want to have good quality data, right?
Yeah,
it's
exactly. I would agree. It's a bit more time consuming and possibly more expensive, but your data is much more robust and believable.
Yeah.
And
Mhm.
sorry, go ahead.
Go ahead.
There's another question, but you were going to say something continuing that theme. Okay. Well, there's another question about can you use the negative control for the double positive CD4 and CD8? And again, I would say no, because that's not the correct control. I mean, you won't be able to define what is positive with the negative alone.
Let me see.
You need to have the two FMOs, the CD4 and the CD8, to be able to do that.
Right. So, you could be tempted here, for instance, to pull it down, right, in the unstained. You see that we're not capturing the spread. I mean, we do have here a bit of, the probably dead cells. I don't know. Let me move this gate.
Well, we don't have a dead cell marker in there, do we? So.
Oh, right, exactly. This is the unstained. Yeah.
So those are a few autofluorescent dead cells sneaking in there, I think.
Exactly. And you could be tempted to do this, and you're not capturing the spread, right? Here, it's not a big deal for the CD4 because we don't have that much spillover. And actually, that's a nice thing of analysis that I remember. So this was Marie Hymas from Cytec. She's been so a proponent of comparing your unstained with your single stain controls and with your fully stained, right? To see how the spread of the negatives change. And actually that's done when you're troubleshooting your panel, right? So if you compare all those things, once you establish your panel, you'll be minimizing these kind of issues, right? And again, like Kathy was alluding to, there's a lot of things that you need to do before you actually reach the analysis phase, and make sure that everything is done with best practices, right? So understanding how staining works. Derek mentioned, washing the samples, probably more than once when you stain them to just reduce all that background signal and to make sure that what you're seeing is actually correct. If you do all these things, the analysis just becomes so much simpler. Okay, so we were looking at this data file, right? But then I started looking at the others, and you start seeing, oh wow, look, there's a population here. Is this double? Are these double positives, or the negative? Where do I set the gate? Again, we need to look at the FMO controls. So I'll go to the. What is this? The FMO control of the. So this is the FMO control of the CD25 positive. And again, it would be nice to have them all appropriately labeled. And so what I'll do is I'll just draw a gate here. And if you're in doubt where, okay. I thought I had drawn a gate, but apparently I didn't. There we go. So this would be the CD25 positive. And the parent gate is CD4, right? So this is all good. We can leave the color like this. And again, I don't know exactly how much to the right. I mean, I'm seeing this population here. So maybe I'll put it here, but I'll use the FMO control for the FOXP3, which is this one, which will enable us to set the gate. So I can just quickly do something like this. I'll set the gate actually right here. Oh, it's so annoying. There you go. I always have to do it like twice or something. Sorry, Fox P3 positive, right? And let's just use another color in case we need to go back to it. Okay. And right, so we can leave it here, and I'm good with this gate here, right? And then now we can go back to the samples that we're seeing, and as you see here, you could conclude. I don't know if you can see this properly because of the color. Maybe I chose the not the best color here for you guys to see, so I'll just change it very quickly again. Let's just use a light blue. Okay. Oh, interesting. This didn't change color. Okay. Anyway, but it's 7.95%, right? And now probably one of the questions you're asking is, okay, so this is the single stain. Sorry, this is the single positive population for FOXP3, but it's negative for CD25. Now, it's very important to understand that whenever we set the threshold between negative and positive, all we can say, especially if they don't resolve, I mean, if they resolve very well, then it's obvious. You have X amount of positives, X amount of negatives, just like here. I mean, I'm sure that there's 14.63% or 14%, let's say, of CD4 positives, and all of these are negative, right? Double negative. Now, when we're looking at these, we can't say that these are positive, these are negative for CD25. It may be just that the levels of expression of CD25 in these cells is not high enough. And since the fluorochrome we're using is not the brightest fluorochrome, actually this is rather dim fluorochrome, we may not be pulling up the whole population. So whenever we set gates that are cutting a population in half, all we can say is, well, we have at least, and let me just, draw a gate here for you to, these would be the, let's say, the double positive, right? So I'll just put CD25 positive, Fox P3 positive. All we can say really is that we have, and I'll join these gates, to make sure that we're, getting all of them. All I can say is that we have at least 8.4% double positives, but I can't say that we have exactly 8.4 positive, right? So this is, I hope you understand, this is a very important distinction, because again, you have to look at your data and realize that you are not resolving the whole population. And this is nice because, yeah, we would expect FOXP3 and CD25 to be doubly expressed. So most likely most of these cells are actually double positive, but we can't resolve them. So this is a limitation of our panel, of our instrument. So all we can say is we have at least 8.4%. Now, what is the problem with this? There isn't a problem with that. You're just not resolving as well as you would wish. But as long as you apply the same strategy, the same gate to all of your samples, again, it's a systematic error, and you're comparing sample to sample, right? This is not an absolute value. It's a relative value that you're going to compare with all your samples. So for instance, here we have at least 5.86%. Here we have at least 7%, et cetera, right? And there we go, 8.42. And then we go to this one, and this one, in fact, as you see, it's negative for FOXP3, right? For some reason, I don't remember what's the difference between this sample and it almost looks like an FMO control for FOXP3.
Well, I think actually that might be the sample that there was no compensation applied to. So that might be why that one looks a little bit wonky. And that's a great takeaway from this, to really make sure that the compensation is applied to all, because if not, that could have a pretty big impact on your files.
True. Yeah, good point. And in fact, it's probably one of the reasons why it's displaying CD8 here, and not the fluorochromes, and the others applying the fluorochromes, because it could be the compensated parameter that FCS Express is interpreting, right? So again, it is important to label everything in the same way, so that then when we're displaying things, we don't have these kind of problems, right? But yeah, good point. So we still have some time. I can show you a little bit of statistics.
Yeah, well, I've got a question here
Yeah.
in the chat actually, which is quite a useful one. And in what type of plot would you recommend for publication?
Right.
I can have a go at that.
Yeah. No, go ahead. Yeah.
Well, what I was going to say, I always say to people, you're in control of your experiment from beginning to end. You can choose how to design the panel, you can choose how to run it, you can choose how to analyze it, and you can choose how to present it. And as we've seen, both FlowJo and FCS Express have different ways of displaying the data. So either histograms or bivariate plots, and there are different types of bivariate plot. And you could use any of those. However, it's important to be aware that there are some publications that have some guidelines about that. So if you want to publish in the Nature Publishing Group, which I'm sure you all do, they would recommend that all your bivariate plots are either like we have on the screen there, the sort of polychromatic dot plots where we can see all the events and we're getting some density information, or that they're contour plots, but also show the outliers below the first contour. Because with both of those, we can see all the data, and the interpretation should be pretty similar. I don't know if you want to add anything to that, Rui, Kathy?
Right. No.
I think.
Yep. It sounds
Yeah,
sounds good, right?
Yeah, it absolutely does. And we also had another question come in from Bucket saying that asking if compensation controls should be applied to all FMO controls exactly the same way, without excluding the color that is missing in the panel. So it's important to make sure that that same compensation matrix is applied across all of your samples, that the panel remains the same. You can have multiple panels that you're running that are going to have panel specific compensation matrices, and that's definitely something that you can utilize, but you would not change the compensation matrix due to the removal of one fluorochrome. So you don't want to do that. And I just wanted to put in my 2 cents from a conversation from a little bit earlier about FMOs and what samples to use them for. When you're developing a panel and starting out, typically you would recommend to do an FMO for each and every single marker that's in the panel in and of itself, right? So you can make sure that you have enough cells at the onset to really test everything out. And then you might wind up identifying that there are primary markers in your panel that are very easily able to distinguish those markers. And if sample quantity is a concern for you, or cell number is a concern for you, that you can then say, all right, for markers X, Y, and Z, I don't need an FMO moving forward, because it's able to really easily resolve. But then there are going to be markers that are expressing at a continuum that you're absolutely going to need every single time to make sure that you have an FMO for. So onset of a panel, FMO for each and every single marker. And then moving forward, you might be able to pare it down a little bit based on how those populations resolve.
Yeah, exactly. I would absolutely agree. And as Rui said earlier, you have to use your cells for the FMOs. So if you can knock a few of those out by looking at the pattern of expression, then that's going to help you.
Right. Yeah. And again, reinforcing the fact that the FMOs help you to troubleshoot actually the panel. Like there are some fluorochromes that are spilling over so much into a certain channel, and if you use that FMO and that spillover is eliminated, that was the culprit, and you can identify the fluorochrome that was actually causing the problem. And so you can then go and just redo your panel, choose another fluorochrome. Maybe that's not as problematic.
Yeah, and then we do have a question that came in from B-Ling, and this is one it's always a very difficult one, and everyone always wants to know a solution. How do you handle multicolor assays when your sample volume is limited and the samples are very precious? Oh, it's a hard one, I think, for all of us.
You handle them as carefully as possible. No, yeah, you might have to sacrifice some things, like maybe you can't wash them, twice or 3 times. if you had more cells, you could do that and maybe improve a bit the quality of your separation. But if you're going to be losing cells and you already have a precious sample and very few cells, you may not have the luxury to do that. So that may compromise the quality of your data. But yeah, you have to I would say troubleshoot with other samples and then.
Yeah, it's a balance, isn't it, between getting good and robust information and not losing too many cells. So you have to weigh up which is going to be the most important to you.
Yeah, exactly. So I would recommend maybe trying with other tissues where you have more cells. Again, this is going to be very different because the titer of each antibody may be different. But you can try to see, look, maybe the tissue is not that different, and just do all the tests, with the washes and the panel, and try to optimize the panel, with a more, controlled sample that you have more cells with. And then once you want to start the experiment, you have most of the things troubleshooted, understanding that there will always be some differences between different tissues, and then just working, as carefully as possible, not to lose cells. Yeah.
We've got another question here. That's unless you were going to go to that one, Kathy, or did you have something to add? Sorry.
Nope. I was going to read that same question.
Okay. I'll leave that to you then.
Okay.
It's a good question.
It's a great question. I know I have my own opinions on this. So we have someone that asked if you're doing a series of experiments using the same panel, would it be appropriate to move the gating and analysis to ensure populations are included? And if so, how would you try and avoid including unconscious bias or objectivity? So I could throw in my 2 cents on it and see if everyone's, all right. So the important thing is there are going to be differences in the instrument and the samples and potentially the reagents from day to day. So if you're running the same exact experiment or the same panel on different days, if that's how I'm interpreting this question, you want to make sure that you're running those appropriate controls each and every time. And it's important to run the unstained, the single colors, the appropriate FMOs from time to time. And there might be differences and in day to day in what those populations look like. That again could be because there's some biological difference and you want to account for that and make sure that you gate for those populations, and that's okay to do. There might be differences in the instrument itself. So if you need to make sure that you're comparing day 1 to day 10 on an instrument, you can't just turn the instrument on, run your sample, and assume everything is going to be comparable. You'd have to run some type of standardization beads to be able to compare data from different time points. But you can absolutely adjust gating as long as you have the appropriate controls in place from day to day to make sure that you're around the positive populations or negative, whatever you're looking for, and make sure that you're gating based on those controls and taking into account any differences in the instrument from day to day.
Yeah, I would pretty much agree with that. Yes, you have to you have to have the knowledge of what you're doing and understanding why you're doing it. So certainly with a longitudinal series of experiments, making sure that the machine is operating the same day to day is extremely important. But as Kathy said, as long as you have the right controls on the right day, there's no reason why you can't move a gate. What you don't want to be doing is sort of changing your gates between the same sample on the same day, for example.
Right.
Yeah, and also too, like, when people are carrying out longitudinal studies, what's your question? Are you comparing population percentages and that's it? Because then you don't necessarily need to run standardization beads. Are you trying to compare MFIs from day to day in population percentages? That's a very different comparison that you're trying to do, because if you're trying to say this percentage of my population is GFP PE double positive on day 1 versus day 10, just the biological control should be sufficient. If you're trying to say I have the MFI of 50,000 on day 1 and 20,000 on day 4, and there's a this X-fold difference, you need to run standardization beads to make sure that the instruments the same from day to day, or we're accounting for changes.
Mm-hmm. Yeah, I usually say that you need to be objective when you set the gates, and then you have to use this objectivity in a consistent way, right, across all your samples for that day. I think one of the important things is also to, if you want to avoid a bit that, variation between instruments, between days, because even the sample preparation done on different days may vary slightly, right? Again, you may not wash as well the cells, or the cells may not be as well washed as you would wish. There's some variability in the centrifuges and, pipetting, all these things, right? So there could always be some variances. So I prefer to, for instance, that's why I was talking so much about the scatter and the, in this case, it's the live dead versus scatter, to try to, let's say you're looking at lymphocytes, then if you want to compare percentages, you want to compare to a population that's always obvious, right? So if it's not very obvious in the scatter, maybe at least, so here, if you're interested in the FOXP3, CD25, etc., T-regs, then you want to express this as percentages of CD4 cells, because as you saw, CD4 cells are easy to identify, regardless of whether we're putting things here or not. Of course, if you include the dead cells, you're going to be skewed, but you're pretty safe here in moving this gate, and the percentages are not varying that much, right? So this is a robust measurement, the CD4 positives, because they pull out pretty nicely. So you do want to use pan markers. That's one of the reasons people don't just label FOXP3 or CD25 and not CD4, and sometimes even CD3. So having these markers just to have very distinct populations that then you can compare them to, I think is important to eliminate all that variability, right?
Yeah, I think it's important to realize as well that the variability, as you say, Rui, it's involved around your sample preparation as well.
Yeah.
Sometimes the pH of your buffers will vary. I probably use fresh formaldehyde every time if I'm making it myself, rather than new stuff that's been hanging around.
Right.
And that all of those things can have an impact on your results.
Right. Okay.
There's also another question that actually came through from Aman asking about, when we're using FMOs, does the intensity of the fluorochrome matter? Should they use a strong color like PE? So I think they're wondering if you should only use FMOs based on the intensity of the fluorochrome.
No, no, it depends on the levels of expression also. So you can have a very bright fluorochrome, and usually it is important to use a bright fluorochrome when you have antigens that are low expressed. But on the other hand, it may not pull out entirely. So, and it depends also on how it is expressed, because some markers are expressed continuously. There's a continuous levels of expression. So there are cells that express less, more, and much more. But there are markers that are just, more homogeneously expressed, and they pull out very well. So depending on all these factors, you want to use an FMO control regardless. Like you said, Kathy, right in the beginning or earlier, that you want to start by when you start your panel, first experiments, you want to bring all the FMO controls, and then once you start getting confident that okay, this always pulls out so clearly, I don't need an FMO control, then I can start dropping those controls. But you really need to be sure. And again, I go back to the clinical samples, right? When you were dealing with an outbred population like humans, you're going to expect a lot of variability. So maybe the first experiment, things work very well. You don't need that FMO control. But the second experiment where you're looking at other patients, you may realize that you need it. Or your fluorochromes, your tandem dyes may start degrading, and suddenly an experiment that, an FMO control wasn't important now can become important. So, I would be also very careful in dropping those FMOs unless you're really confident that you won't need it.
Be vigilant. Keep watching the skies.
Exactly. Okay, so we're running out of time, and
we are. And
this is the part that I don't remember as much in this software, so I'll try to go fast without making too many mistakes. To do well for reporting and statistics. Actually, the software is relatively simple. So if you select the gate and you right click, you can just say go to statistics and create gate statistics. And the nice thing is, regardless of the gate, it will open up a dialog. Let's put it here, you know, with a lot of statistics for all of the gates, right? The cells, singlets, CD8 positive, CD4. So for instance, if you don't want this gate to, you know, this was just the back gating, you know, we can go here and where is it? Like gates to display. You can actually, so here we're telling display all gates. We can just say, no, we want, oh, sorry, all gates except the ones that check below. So we can just go like this and then we eliminate it, right? If you don't like, so I don't know why the geometric mean is displayed on purpose, by default, sorry. But you can, you know, click here statistics and just say, well, I don't care about the geometric mean. I'm just looking at the percentage of events, of gated events, or of all cells, right? And the number of events. So you would have this here. Again, you could play around and put whatever you add, whatever you want, the medians, etc., standard deviation, whatever you want to display, you could display in this statistic view. In terms of batch processing, right? Because now the thing is, you want to start looking at all these, making sure. I like to go through all of the samples and just to make sure that the gates make sense, right? Because if they don't, for instance, here, this seems very different. Now, is it real or not? But as Kathy pointed out, this is the sample that doesn't have a compensation matrix applied to. So most likely, we don't have here proper compensation applied. So we probably should kind of discard this sample, right? So let's imagine that you want to now do batch processing. Let's say you want to now look at all these files in PowerPoint, right? You want to make, you want to show this in your lab meeting or something like that. Okay, so we go here to batch, pretty obvious, and we start with the batch actions. So what do we want to do? Okay, oops, we want to save to PowerPoint, right? So I'll just click it here, and then okay, include background. This doesn't matter. Let me see. The quality could be for on-screen or for printing. Combine the output from all iterations in the same presentation. This is what we want to do. We want to have all these things in the same presentation. And start with an empty file, and we're going to save it to, let me see, I don't know where this is, but I think exactly, it's in the right place. So we'll just call it PowerPoint or PPT of my data, right? And you can click ask for confirmation before overwriting an existing file, right? If you're concerned about overwriting files, etc. And we'll leave it as such. So we created an action, but we haven't processed the batch. And actually, before doing that, I actually, what I'm going to do is just place this over here so that I have my page. And now what it's going to do, it's going to run through all of these and show each page with a different file. Do we want the FMOs? You may want the FMOs just to check to see if they make sense or not. You could eliminate them. Now, again, we could delete the files that we don't want, or we can just go here and I'm going to say, yeah, let's try all the FMOs. So I'm going to click here because it will start in the active file, right? So I'm going to click here. And actually, if you don't know, let me see, if you go to, I think it's review, would you like, there's no pause options? Yeah. Right, it's already saying, so it's going to automatically perform the maximum as possible, currently 17. You could say, well, no, just do the first 5 files, right? Or you can just run through all of them, right? So this is a way to see how many. Now, if I click here, I don't know if it's going to, no. So if I click here, for instance, let's say I just wanted the samples, right? I would click here and I 1, 2, 3, 4, 5, and I could go now to review. I never know if it's slow or if it's just not not doing anything. And there you go. Currently 7, 1, 2, 3, 4, 5, 6, 7. But we can say no, just do 5, and it would do only the first 5. right? So we could do that, but let's do all the FMO controls and just have a little bit of fun here. Oops, I could cancel. Never mind. And let's do the 17 iterations. And yeah, hopefully it's not too slow. And now we can go ahead and just run the batch. And we just say yes. And there he goes. Okay, not too slow. Now, the nice thing is that we could actually go through all of them and ask it to pause. So do you see that the data is changing here? The data is changing as it's doing the iterations. So we could actually ask it to pause and we could verify the gates. They look good, continue. And we could do this, you know, the iterations semi-automatically, let's say. Okay, no errors, this is perfect. Okay, now let's go and see the result. There we go, PowerPoint of my data. It's already writing it to the cloud. And time is ticking.
So as that time's going by, we actually have a question from one of our attendees that's asking what the purpose of this step is for doing this batch analysis.
The purpose? So the purpose is for, you know, there are several purposes. One of them is if you want to, for instance, now present your data and show all your data. Do you see here? Now each page is a unique file with the same gates applied, right? Where, and again, like Kathy mentioned in the beginning, you can even do data specific gates. It would respect that. And you'll just have all the files where you can, for instance, check the, you know, for the FMOs, I think this is the FMO for the CD8 and check to see if the gates make sense. And you can see all the statistics for, you know, all of these files, right? Now, the thing is, what if you want to export this to, so this is the other purpose of doing the batch analysis. What if you wanted to export the percentages of FOXP3 for all of the files, including the FMOs, let's just say, you know, or yeah, let's just say for all the files for simplicity, but you could do it just for your data files. So in this case, we would go to, you know, we would add a report, and this would be, right, let's call it just report, right, that will be enough. And we'll try to see if it opens because it wasn't working for me. And so, oops. So we have a report. Now we need to add statistics. Okay, how do we add statistics here? This is so slow. There we go. And we can, one of the nice things here in FCS Express is we can actually add a spreadsheet. Yeah, let's call it a new spreadsheet. We can add a spreadsheet here, right? And we can ask the, you know, the software to display, you know, to display statistics. So let's say I want to see the percentage of CD4. First, I want to make sure that I have the name of the file. So I'll drag this over here, and let's say, and it's going to ask, so what do you want to write there? And I'm going to say, I want a token, which will be a keyword. Now you could check all the keywords of the files. I know by heart that the file name is something like this. And so I'll add the file name. And so it should add, there you go, the name of the file. And then I can, you know, in this cell, I can add, for instance, the C4 percentage gate, or actually we were looking at the double positive, right? So let's drag this. Oh my God, this is a bit slow. Drag this here, and it will automatically, oh, actually it won't automatically. So here we can ask, okay, I want the percentage of gated cells, right? Of the double positive, and there we go. And 5.64. Now, what if I click another file? You'll see that it refreshes, right? What if I click on this file? You'll see that it refreshes. And so it's always refreshing the name and the statistics. Now, what is this good for? Well, what if I then decide, okay, now I want to see, I'll drag these guys over here. and now I can add these statistics, and I'll click this one also, and I'll have them in different columns, and the iterations will be in the rows, right? So I'll do the same here, and I'll do just very quickly. So I'll go to properties. So this will be the percentage. So if I remember exactly column name, so we'll call this the Fox P3 positive, CD 25 positive, right? So I'm almost done with this. And here we'll do the same. This will be the file name, right? So I'll go to field and just call it file name, right? And now, hopefully, when I run this batch, so I'm not going to do the PowerPoint again, so I'll unclick it, and I'm going to run the batch. Remember, this is, it's starting here with this sample, so I have this sample selected, correct? And so it will do for all of these samples, right? Just for the sake of simplicity and time. And so now when I go to the batch processes and I say run, and you'll see it starting to change, and it's changing that. And we should see, well, it should open the Excel automatically. I don't know if it will, but if it doesn't, I'll just go here and open it. There we go, reports, and I can open the Excel sheet, and I'll have all my statistics that I can then report and do my hypothesis testing. Now you could run the mean and compare it with the with the treated samples versus the there we go. So as I showed, I renamed the column names. So we have the file name and we have the percentages of double positive for FOXP3 and CD25. And you could do this for many other things, right? You could add more columns with other statistics. You could add more files. You can separate in, and then you could use pivot tables here to do your analysis. And again, do the hypothesis testing on your data, right? And actually, this is this is an Excel file, but you could actually export it to a CSV if I'm not mistaken. Right, no, in this case, it's only an Excel file, but you could export that into a CSV file and use R to do statistics on it or anything else. So, so it's basically, like I said, FlowJo also does all these things in a different way, but there should be always a way of doing this very automatically. Imagine you have hundreds of samples or even 100 samples. You're not going to write down the statistics by hand. You can just do this and you would export. It doesn't matter if you have 2 or 200 samples.
Thanks, Rui. We should probably think about wrapping up now.
Mm-hmm.
Can I just share my screen, because I've just got a couple of last slides just to let everybody know what's happening next. There we go. Right. Okay, so thanks everybody for coming. As we said, we have recorded this webinar, so the recording will be made available hopefully Monday, Tuesday, or Wednesday next week. And all of the recordings of all of our webinars are here on the OpenFlow channel on YouTube. Hopefully we'll also send you a questionnaire. So if you've got any things you want us to address over the next few months, please do let us know. We will take a little bit of a break for the summer, but we are going to have one more data analysis session before we do that. And that's going to be 4 weeks from today on the 23rd of July, same time, same place. And we'll look at compensation, offline compensation. We'll use probably both FlowJo and FCS Express to do that. And we'll look more into the idea of data spread and what effect that has on our results and our data. So you can register for that already, this very complicated URL here, or if you just go to the crick.ac.uk what's on page, you can register for that. But we will also put that onto Purdue, LinkedIn, and everywhere else over the next few weeks. So the very last thing to say is you can contact us. You can contact us individually or by using the OpenFlow Cytometric gmail.com address. that will come to all of us, and you might even get 3 answers if you're lucky then. And of course, you can always follow us all on Twitter as well. So thank you all for coming. Hopefully we've answered all of your questions. So if you do think of anything else, please do contact us. But that's it for today. Have a good afternoon or evening wherever you are, and hopefully we'll see many of you in a month's time. Thank you all for coming.
Thank you, everyone.
Bye.